Each hemoglobin molecule is composed of four polypeptide chains, each with an iron that contains a heme group. So each hemoglobin can transport four oxygen molecules. When hemoglobin is bound to oxygen, that's called an oxyhemoglobin. If the hemoglobin has released the oxygen, that's called a reduced hemoglobin or deoxyhemoglobin. Loading and unloading of oxygen is facilitated by the change in shape of the hemoglobin. When oxygen binds, the hemoglobin is going to change shape, and that's going to increase its affinity for more oxygen. If the oxygen or when the oxygen is released, the hemoglobin is going to change shape again, and that's going to cause a decrease in the affinity for oxygen. When the hemoglobin is fully saturated, that means that all four of the heme groups are carrying oxygen molecules. If it's partially saturated, that means that only one or three of the hemes are carrying oxygen molecules. The rate of loading and unloading oxygen is regulated to ensure that there's an adequate amount of oxygen being delivered to cells. So the factors that influence hemoglobin saturation are partial pressure of oxygen. Other factors include temperature, because increased temperature allows for more oxygen to be released. Blood pressure, pH. The lower the pH, the more oxygen is released. Uh, Partial pressure of carbon dioxide, and the concentration of BPG. So BPG is it's a molecule. So it's a biphosphoglycerate, which is the molecule that helps hemoglobin bind to oxygen. When the partial pressure of oxygen um, is binding and releasing oxygen with hemoglobin, the percent of hemoglobin saturation can be plotted against the partial pressure of oxygen. This results in an S-shaped curve, and it's referred to as the oxygen hemoglobin dissociation curve. And so the way that the partial pressure of oxygen influences hemoglobin saturation can be seen in, in blood vessels. So in arterial blood, the partial pressure of oxygen is 100 millimeters of mercury, and it contains 20 milliliters of oxygen per 100 milliliters of blood. Here, the hemoglobin is 98% saturated. When you increase the partial pressure of oxygen, like what happens when you take deep breaths, that's only gonna produce a small increase in the oxygen binding. And we're gonna see that on the graph that's coming up, showing the S-shaped curve. And in venous blood, the partial pressure of oxygen is going to be 40 millimeters of mercury. And here the hemoglobin is going to be 75% saturated. So here's the S-shaped curve on the graph that shows the amount of oxygen that's carried by hemoglobin and how it depends on the partial pressure of oxygen. Because depending on whether it's a high partial pressure of oxygen or a low partial pressure of oxygen, that could dramatically change the percent of the oxygen saturation of hemoglobin. So if we look at this top graph, we can see that the partial pressure of oxygen is at 100 here on the far right. And here, at this point, the percent of oxygen saturation of hemoglobin is at 98%. But if you move that partial pressure of oxygen down to 80, the saturation of oxygen on hemoglobin is only going to be reduced by 3 percentage points. So it'll go from 98% saturated to 95% saturated. So up here you can see that at high partial pressure, the hemoglobin curve is almost flat. So it shows that the hemoglobin it's pretty much saturated with oxygen at this point. But when we go to a lower partial pressure of oxygen, like at 40 millimeters of mercury, here the saturation is going to be at 75%. And if we go from 40 to 20, there's going to be a big change in the saturation of oxygen of hemoglobin. because it's going to drop down from 75% to 40%. So it's a here at low partial pressures of oxygen, large changes in the partial pressure are going to cause large changes in saturation. But at high partial pressures of oxygen, large changes in in the partial pressure only cause small changes in the saturation. The influence of other factors on hemoglobin saturation are are things like temperature, hydrogen ions, the partial pressure of carbon dioxide, and the BPG molecule that can modify the structure of hemoglobin. So it results in a decrease in the hemoglobin's affinity for oxygen. So this occurs in the systemic capillaries. And it also enhances the oxygen unloading, and this causes a shift of the oxygen hemoglobin dissociation curve um, to the right. So the curve moves slightly to the right. And the decreases in these factors will shift the curve to the left. So this decreases the oxygen unloading from blood. So moving to the right would just mean that the curve would slightly move this way. And to the left would just be to the left of this one here. So shifting to the right just means that the hemoglobin has less affinity or a decreased affinity for oxygen. And moving to the or shifting to the left would just mean that it has an increased affinity for oxygen. Here you can see the temperature effects, the partial pressure of carbon dioxide, and the blood pH, and how they affect the oxygen hemoglobin dissociation curve. Well, really here what you can see is the temperature, because if this would be the normal body temperature at 38 degrees Celsius. If the temperature increases to 43 degrees Celsius, then the percent of oxygen saturation of hemoglobin is going to decrease with increased temperature. If you lower the temperature to 20 degrees Celsius, then the affinity of oxygen to hemoglobin is going to increase. Other factors that can influence the hemoglobin saturation are BPG, or the BPG molecule, which is again a molecule that helps the hemoglobin bind to oxygen. So this is produced by the red blood cells during glycolysis, and BPG levels rise when oxygen levels are low. Then there's also the declining blood pH and increasing the blood um, partial pressure of carbon dioxide. This can cause the hemoglobin oxygen bond to weaken, and it's referred to as the Bohr effect. Heat production in active tissues also directly and indirectly decrease the hemoglobin affinity for oxygen, because the higher the heat, the less affinity that the hemoglobin will have for the oxygen. And this allows for the increase of oxygen unloading to active tissues. Carbon dioxide is transported in blood in three forms. It's dissolved in plasma, it's chemically bound to hemoglobin. So when it's bound to hemoglobin, it forms a structure that's called the carbaminohemoglobin. But the vast majority of the carbon dioxide is transported as bicarbonate ions in the plasma. So this formation of bicarbonate involves the carbon dioxide combining with water to form the carbonic acid, and this dissociates into bicarbonate and hydrogen ions. So at lowered pH, the hemoglobin would release the oxygen at the tissues. So here's the hemoglobin oxygen structure. It would release the oxygen, and then that hemoglobin would bind to an excess hydrogen ion in the red blood cell. Then the bicarbonate ions, they would move to the plasma, out here to help buffer the other hydrogen ions. In the pulmonary capillaries, the CO2 is getting pulled into the lungs where the CO2 levels are lower. So here's the alveolus of the lungs. And bicarbonate moves into the red blood cell. So this bicarbonate is going to move into the red blood cell where it's converted into carbon dioxide and water. Changes in respiratory rate and depth affect the blood pH. So slow, shallow breathing causes an increase in carbon dioxide in blood, and it results in a drop in pH. The rapid, deep breathing causes a decrease in carbon dioxide in blood and results in a rise in pH. So changes in ventilation can help adjust the pH when the disturbances are caused by metabolic factors. So breathing plays a major role in acid-base balance of the body. Hypoxia is when you have an inadequate oxygen delivery to tissues, and it can result in cyanosis, and it's classified by different causes. So if it's anemic hypoxia, that's when there's too few red blood cells, or if there's too little of the hemoglobin. Ischemic hypoxia is when there's a block in blood circulation. And then carbon monoxide poisoning, which is something that happens, especially when there's like a fire, is when the hemoglobin, or it's caused because hemoglobin it has a higher affinity for carbon monoxide than it does for oxygen. So it prevents the oxygen from binding to the hemoglobin. All right, so that's going to be it for chapter 22, and I'll continue on the next chapter.